Now we're going to discuss different excretory systems. Central to homeostasis, excretory systems dispose of metabolic waste and control solute concentration. Excretory systems produce urine by a two-step process. First, the body fluid, blood or hemolymph, is collected. Second, the composition is adjusted by selective reabsorption or secretion. Filtration is the initial fluid that is passed through a selectively permeable membrane of transport epithelia. Blood pressure forces liquids and small solutes through the fluid that passes through. This is the filtrate. Excretory systems use active transport to reabsorb solutes from the filtrate. There are different types of excretory systems. First, the protonephridium is found in the flatworm. It is a network of dead-end tubules lacking internal openings. The tubules branch through the body and end in flame bulbs. That is a tuft of cilia that goes inside the tubule. The cilia beats to draw in water and solutes. Urine exits the tubules through openings called nephritiopores. Metanephridia are found in annelids, segmented worms like leeches and earthworms. They have internal openings that collect body fluids. Each segment of the worm has a pair of metanephridia. The opening of the metanephridia is surrounded by a ciliated funnel, the nephritosome, which collects fluid from the coelom, or the body cavity. Since they live in moist soil, they take up water. Urine in the tubules reabsorbs solutes to the blood. Waste in the tubules is dumped to the outside in dilute urine. Malpighian tubules are found in insects and other terrestrial arthropods. They remove waste and osmoregulate. They open into the digestive tract and dead end at the tips, which are bathed in hemolymph. Hemolymph has solutes or waste that go inside the tubules, then to the rectum. Water and solutes go back into the hemolymph, and solid waste leaves as dry matter. This type of excretory system is very effective at conserving water. It is one of several key adaptations that contributes to arthropod success. The vertebrate kidneys osmoregulate and excrete. They are made of tubules. We will focus on mammal kidneys, especially humans. Mammals have a pair of kidneys. They are bean-shaped and about 10 centimeters or 4 inches long. The blood supplied to the kidney is by a renal artery, and it leaves through a renal vein. Very large amounts of blood flow through the kidney. The ureter is a duct where urine exits the kidney. One comes from each kidney and empties into the bladder. The bladder stores urine. The urethra is a tube from the bladder to the outside. Sphincter muscles near the junction of the urethra and bladder control urination. There are two regions of the kidney, the renal cortex, which is the outer region, and the renal medulla, which is the inner region. In both regions, there are microscopic tubules and blood vessels. The nephron is the functional unit of the kidney. A long tubule and ball of capillaries called the glomerulus make up the nephron. The blind end of the tubule forms a cup-shaped swelling called the Bowman's capsule. It surrounds the glomerulus. Each human kidney has about 1 million nephrons, totaling a tubule length of 80 kilometers or 50 miles. Blood pressure forces fluid from the blood into the glomerulus and then into the Bowman's capsules. The filtrate in the Bowman's capsules contains salt, glucose, vitamins, urea. It is in concentration equal to the blood. The filtrate passes through three regions: the proximal tubule, the loop of Henle, and the distal tubule. The distal tubule empties into the collecting duct. Many collecting ducts empty into the renal pelvis, which is drained by the ureter. About 80% of the nephrons are cortical nephrons. They're confined to the renal cortex, and they have short loops of Henle. 20% of the nephrons are juxtamedullary nephrons. They have long loops of Henle, and they extend into the renal medulla. They are only found in mammals and birds. This enables them to produce urine that is hyperosmotic to body fluids. It's very important for water conservation. Nephrons and collecting ducts are lined by epithelium that processes the filtrate to form urine. It reabsorbs solutes and water. 1,000 to 2,000 liters of blood flows through the kidneys each day. This is 275 times your total blood volume. The nephrons and collecting ducts process 180 liters of initial fluid, which is 2 to 3 times your body weight. Of this, all sugars, vitamins, nutrients, and 99% of the water are reabsorbed into the blood. This leaves only about 1.5 liters of urine to be void. When we look at the blood vessels associated with the nephrons, there are the afferent arterioles that supply blood to the nephron. They branch off of the renal arteries. They branch into capillaries of the glomerulus, and they converge into the efferent arteriole as they leave the glomerulus. The efferent arteriole subdivides into the second network of capillaries, the peritubular capillaries. They surround the proximal and distal tubules. Additional capillaries extend down to form the vasa recta. These are capillaries that serve the loop of Henle. All the tubules and capillaries are immersed in fluid that help with diffusion between the capillaries and the tubules. If we look at blood to filtrate a little bit closer, we'll start with the proximal tubule. Secretion here is the movement of substances from the blood to the filtrate, and it reabsorbs. Its reabsorption alters the volume and composition of the filtrate. For example, pH is controlled by the secretion of hydrogen ions. The proximal tubule reabsorbs 90% of the buffer bicarbonate. Drugs are processed in the liver. They pass from the peritubular capillaries to the interstitial fluid. They're secreted across the epithelium of the proximal tubule into the nephron. Nutrients go from filtrate into the interstitial fluids, then into the peritubular capillaries. One of the most important functions of the proximal tubule is the reabsorption of salt and water from the filtrate. When we look at the descending loop of Henle, it is responsible for the reabsorption of water in addition to the proximal tubule. The epithelium is permeable to water, but not salts and other solutes. As the filtrate moves down towards the medulla, it loses water. In the ascending loop of Henle, filtrate moves back towards the cortex. It is the opposite of the descending loop. Epithelium is permeable to salt, not water. By losing salt without giving up water, the filtrate becomes more dilute. In the distal tubule, another important site of secretion and reabsorption, it regulates potassium and salt concentration. In the collecting ducts, the filtrate is carried through the medulla to the renal pelvis. This also controls salt concentration, absorbs water, and the filtrate becomes more concentrated. In the inner medulla, the duct becomes permeable to urea. Urea diffuses into the interstitial fluid. There's a high concentration, so more water can be extracted from the urine. Along with salt, this interstitial urea is a major solute contributing to the higher osmolarity that enables the kidney to conserve water by excreting urine that is hyperosmotic to the general body fluids. The mammal kidney's ability to conserve water is a key terrestrial adaptation. The osmolarity of the human blood is 300 milliosmoles per liter, but the kidney can excrete urine 4 times as concentrated, about 1,200 milliosmoles per liter. Some animals who live in dry environments can excrete urine 9,300 milliosmoles per liter, which is 9 times the concentration of seawater and 25 times the concentration of their own body fluid. When we look at the hormones that regulate the functions of the kidney, one of the most important aspects of the kidney is its ability to adjust volume and osmolarity of urine. Antidiuretic hormone, or ADH, is produced in the hypothalamus and stored and released from the pituitary below the hypothalamus. When the osmolarity of blood goes over 300 milliosmoles per liter, more ADH is released and goes to the kidney. ADH increases the permeability of the epithelium to water, so water reabsorption goes up and urine volume goes down. But only the consumption of more liquids can bring osmolarity back down to 300 milliosmoles per liter. The JGA is a specialized tissue near the afferent arteriole, which supplies blood to the glomerulus. When blood pressure drops, the enzyme renin helps form a protein called angiotensin II. It increases blood pressure and blood flow. It also stimulates the adrenal glands, which sit on top of the kidneys, to release aldosterone, which makes the nephrons reabsorb more sodium and water. This increases blood volume and pressure. The atrial natriuretic factor ANF opposes RAAS, the renin-angiotensin-aldosterone system. The ANF lowers blood volume and pressure.